Hi, my name is Diana Doe, and I am a postdoctoral researcher in Howard Chang's lab at Stanford, and I'm also an NIH NIAMS K99 grantee. So I've always had an interest in biomedical research. I entered actually Anthony Fauci's lab at the NIAID as a post-bac trainee. So I was in the IRTA program, and that was a lot of fun. And so the more I learned about immune pathways and the complexities and the importance of the immune system, the more interested I became in blood cells and development and disease and how these pathways are regulated. And so that's what led me to Hana Miglo's lab at UCLA. That's when I became interested in non-coding aspects of gene regulation. And that's what led me to Howard Chang, who is known as the godfather of link RNAs, non-coding RNAs. Non-coding RNAs are elements that are involved in, I used to think of them as kind of the invisible architects the genomic universe. When I was developing my postdoc project with Howard, I mentioned, I still have a very long-standing interest in immunology, the immune system, and human health. But I was also interested in studying regulation of these immune pathways from the standpoint of the non-coding genome. And so this is how we came to work on the EXIST project together. We found a novel role for EXIST ribonuclear protein particles that helps explain the female preponderance for developing autoimmunity. At the time when this project began, it was kind of novel. And so sometimes when you have a novel project or a novel hypothesis, there's some reluctance for different funding agencies to, in a sense, take a risk on your hypothesis and take a risk on you and your ability to actually see this project to fruition. Yeah, I actually have been sponsored by the NIH for quite a while. I think when I was at UCLA as a graduate student, I was part of a training grant that was sponsored by NIGMS. And now I am a grantee funded by the NIAMS. It's great that the NIAMS and the NIH is out there to fund really novel and cutting edge research